I think it's just cursor position.
It's fine.
Okay, we're going to get started then. Thanks everybody for coming. Today we're really happy to welcome Chris Gilliam from the Department of Material Science and Engineering. He's also an affiliate member of our department, and as you'll see, he does things very similar to many of the things that we do. So Chris began life as a chemist, we won't hold that against him, and actually worked as a chemist in industry for several years. Actually over the time he was getting his master's degree as well. So you've had a lesson, right? You can do many things simultaneously. Then went off to Australia to get a PhD. Returned to the University of Chicago for a postdoc. And along the way somewhere he picked up all this cool stuff about how materials interact with cells, which is what he's going to tell us about today. So Chris, thanks for coming.
Well, thank you, Michael. It's my pleasure to be here. Thanks for the invitation. And so I've spent most of my time since I started my faculty position around material scientists. So it's great to be able to come and talk to the bioengineers because my group does a lot of fundamental biology. And as I'll try to, can everybody hear me? Should I scream louder? I'm testing the acoustics of the room. As I'll try to show you throughout the talk, a lot of what my lab does is it looks at biological systems and is motivated by some hypotheses based on biology to then develop materials and use some of these materials-based platforms to answer some very fundamental biological questions. And as Michael mentioned, I'm a professor in material science, but I'm also affiliated here in bioengineering, so I like being involved with bioengineering. I also have an affiliation in the micro and nanotechnology lab, where I've got a stem cell lab where we do a lot of our culture, and we use a lot of the great fabrication infrastructure there to do a lot of patterning and things of that nature. And I'm also involved with the IGB in their regenerative biology and tissue engineering theme. And so what I think we bring to the table is we are really interested in some of these, how we can really breathe life back into some two-dimensional assays, where there's very talented individuals on campus and all throughout the world that build very beautiful, complex three-dimensional scaffolds. And so we're still at the 2D level, but hopefully some of the insights that we gain, we're hoping to translate into three-dimensional materials for next-generation type applications. Okay, so here's just one of these montage slides of overview of my lab. We do a lot of micropatterning, and the reason we like micropatterning is there's a lot of interesting things you can do when you capture a single cell or two cells or multiple cells in various architectures to study how they signal across junctions, how they communicate with their extracellular matrix that they're sitting on top of, and how we can control that in a rational way such that we can study the role that things like geometric cues and actomyosin contractility play in guiding, ultimately, gene expression, which will tell a cell what to do, whether it's going to hang out or in the case of stem cells, turn into something that you're interested in. We're also interested in nanostructured materials. Biology has a lot of nanostructure in it. So if you look at a lot of the membranes in biological systems and a lot of the hard materials that are already there in biology, often the nanoscale presentation of things like the mechanical environment or the presentation of ligands is crucial for conveying signals to cells to tell them to either migrate, to stay put, to die, to differentiate, all across the board. We're also interested in molecular engineering. As Michael suggested, I am a chemist, and it's true from my early days. And there's a lot of things we can do with surface chemistry, utilizing platforms where we can vary the presentation of cell adhesion ligands and growth factor ligands to see how these can signal to cells to get them to perform certain functions, to understand how they work. We're also interested in hydrogel materials, and so this is where we start getting closer to 3D. And the reason we're interested in hydrogel, just like so many people in this department and others are, is biological materials are comprised largely of polymeric architectures with biopolymers, and so understanding things like the elasticity of the microenvironment or the viscosity of these things is important. And so combining some of these platforms together, I feel, is really exciting because we may be able to develop models that more closely recapitulate the way that cells and tissues form during development. And this will help us understand developmental processes as well as ways that we can design new materials for applications like tissue engineering. All right, so here's an overview of what I'm going to show you today. I'm going to talk about some of this work because I think it builds a nice cohesive picture of how we can use these material science-based platforms to study cells and cell signaling. And so we're going to explore the cues in the microenvironment. I'm going to tell you a little bit about geometry, how the geometry of single cells can influence the cytoskeleton, which can then influence gene expression. I'm going to tell you a news story that's emerged that I think is really exciting, and that is when you study these individual cues, we're often missing the big picture, which is that in vivo, you're not just going to have a cell responding to something like stiffness or a cell responding to something like nanostructure. There's going to be an integration of these cues, and there's going to be a synergy and complementarity that arises, and we're really excited about that. Some of our recent work has shown that biological signaling, so paracrine and autocrine events, can play into some of these pathways. And then finally, I'll talk a bit about some chemistry and how we can modulate stem cell differentiation using by modulating the presentation of adhesion ligands on a surface. And then I'll talk about some of our current efforts in trying to combine these things with this idea that if we can incorporate multiple cues together, we can maximize the outcome that we want, and to more clearly understand how cells do this naturally in vivo, as well as design new materials. And then I'll tell you, a bit of a different story, but using the same techniques, is something that's recently come out where we found that patterning may be able to be used to prolong multipotency in stem cells in culture. I'll show you a bit more about that. Okay, so today I'm primarily going to talk about mesenchymal stem cells. I imagine everybody in this room has heard of these cells. So I'm not going to belabor it, but these are an exciting cell for a number of reasons. Primarily because they're a very good model system for studying stem cell biology experiments, whether that's mechanobiology or differentiation type studies, because they're relatively easy to work with. You can get them from commercial vendors these days, and but in addition, they're also still a pretty exciting candidate for regenerative therapies because of this reason. They can be isolated from bone marrow as well as fat aspirates. A lot of people are using these adipose-derived stem cells, and in fact, the literature is suggesting now that these cells may be a parasite and reside on your vessel walls and could be present in every tissue throughout your body. And so this is exciting for obvious reasons, because these cells, or closely related cells that we can identify with our marker schemes, may be in every single tissue. And they may have slight differences, but they all follow a similar set of rules to get them to regenerate tissues using standard homeostasis processes. One thing that has emerged from the use of these, particularly in clinical settings, there are a number of clinical trials underway for using these, primarily for a trophic response, where these cells are shown to secrete molecules that may promote angiogenesis, for instance. But we've been investigating these, but really at the state that things are at right now, we have a difficult time controlling the way they specify and what they turn into when they differentiate. And so really understanding the microenvironment better so that we can design better materials for therapy and things like this is important. All right, so I like to make things simple because these systems are incredibly complex, as I think everyone understands who's studied biology. So when we talk about signaling in the microenvironment, there's a number of signals that we can consider. Physical signals is one that is important, and as a material scientist, we're interested in exploring some of the physical material properties and how they influence cells. So these are things like the shape of cells, the shape of tissues, how those arise, and how you may be able to manipulate cells that way. The mechanical properties, so stiffness or softness of the matrix, stretch. We're all moving all the time, and our matrices are stretching, and strain is being put on various tissues, and this clearly has an influence on signaling. Chemical cues is my second area that I break things down into, and these include things like small molecules, but more importantly for the purpose of this talk is the precise chemical moieties that you see in the extracellular matrix. So when you have a cell sitting on top of a bed of fibronectin protein, there's a lot going on. It's not just that they're interacting with the protein. They're recognizing very distinct moieties in the ECM. So things like our friend RGD peptide that everybody in biomaterials is aware of. But there's other synergistic ligands and other ones that haven't really been fully explored that have a function. And so we're also interested in exploring what the function of these precise ligands are. And I think there's a lot of room for discovering this because there's so many integrin ligands on the surface of the cell and so many different ECM proteins. And And then the area that's probably been studied the most is biological signaling, as I call it. And so I put into this little bucket things like paracrine and autocrine signaling, growth factors, hormones, etc., as well as signaling across junctions, so coherent junctions and gap junctions, and how this might influence cell fate. And really the theme, I think, that's coming out in the field, and you hear more and more about this, this integration of these cues and trying to understand how they play into one another, I think is really important, because clearly things like shape and stiffness is going to influence the presentation of the adhesion ligands that a cell responds to. And evidence, some of which I'll show you today, suggests that these things can lead to differential signaling of biological factors. Okay, so we'll talk about physical cues first. There's a lot of things that have been done and a lot of studies that I think are important for exploring this space. Nanostructured surfaces, clearly, I mentioned that a lot of biology is nanostructured, but a lot of these techniques we may be able to use to make new tissue engineering scaffolds, for instance, that have this component. In this example here, these are titanium oxide nanotubes that are grown in different sizes. I know it's probably tough to see back there, but essentially, when they put mesenchymal stem cells on these surfaces, they found that cells adherent to these small nanotubes were able to spread out, get a high degree of cytoskeletal tension, and a lot of robust focal adhesion formation throughout the cell, compared to cells that are adherent on these large nanotubes, where they remain rather spindly, and they don't adopt a stable cytoskeletal configuration. And then after several weeks, when they went in and stained for markers associated with bone cell differentiation, they found that cells that were spread out had this healthy, well-developed cytoskeleton, expressed higher levels of differentiation markers for bone. Similarly, you can do similar studies with strain. And so here's showing some mesenchymal stem cells that are being stretched on a polymeric substrate. And by doing this, you can see differential regulation of genes associated with different differentiation outcomes. So here we have alkaline phosphatase activity in cells that were stretched for a period of time, have higher levels of bone cell differentiation, while, and that's true for several other markers, while they see a decrease in genes associated with fat. And so what a lot of these studies are pointing to is that cytoskeletal tension appears to be something that's important for bone cell differentiation. A real seminal work came out of Dennis Disher's lab at Penn with Adam Engler as the first author, who's now at University of California, San Diego, and he's visited us last year, I think. Adam and Dennis published this paper in 2006 that I think really changed the way a lot of the material scientists in bioengineering thought about these things. And what they did is they did a simple experiment by varying the stiffness of polyacrylamide gels, so standard gels used for running protein gels, from very soft gels that approximated the stiffness of brain tissue, so less than 1 kilopascal. And then they increased the cross-linking density of those gels to make them more rigid, to approximate that of muscle tissue and bone tissue. And they made a really startling observation, but which I think makes a lot of sense, in that as you change the mechanical properties of the matrix of which these stem cells are sitting, you change the way that they differentiate. So here you see cells on this soft matrix are expressing markers associated with neurons. They're developing neuronal-like phenotypes too by extending out processes. On the intermediate stiffness gels, you see muscle markers, and finally on the stiff gels, where the cells are very spread, they have high cytoskeletal tension again, they start expressing bone markers. So I think it's a really exciting demonstration of the importance of biomaterials in stem cell fate decisions. Another important story that will play into some of the work I'm going to show you today is this work that came out of Chris Chen's lab, also at Penn, where they explained something that had been observed with some of the early reports of mesenchymal stem cells. Basically, when they first started calling these mesenchymal stem cells, they showed that if you seed them at low density on tissue culture plastic, and then you seed them at high density on tissue culture plastic, the cells that were initially seeded at low density, after several weeks, showed a much higher degree of bone cell differentiation. And no one was really clear why that was. I think a lot of the initial arguments was that clearly there's a paracrine signaling mechanism where biological signals are diffusing across the dish. But what Chris Chen and now others have done is they used micropatterning to culture cells in either small islands where they remained as big balls, just sitting on the surface with very low cytoskeletal tension, or they put them on these large microislands that they patterned on glass, such that they can spread out and get a high degree of cytoskeletal tension. And then they mixed two medias together, one that promotes fat and one that promotes bone. And after about a week in culture, they stain them for lipid droplets, which are shown in red, and they show that cells in these small islands almost exclusively turn into fat, while cells in these large islands that are well spread almost exclusively turn into bone. And so clearly, the spreading of these cells is important for differentiation, and it makes sense in terms of other observations that have been made with other material systems that showed that when a cell is very well spread on a nanostructured surface or on a stiff matrix, and they prefer to differentiate to bone than if they're balled up or rather spindly. They went and did some very nice, elegant work to show that this involved some signaling and pathways that utilized a feedback loop between this row A, small GTPase, as well as row kinase, and that in the absence of high levels of row A, cells would prefer to turn into fat. Okay, and so I think another exciting thing about patterning that we can do, outside of the spreading thing, is we can explore other geometric cues that can influence these cells. And this is a picture that I really like that came out of a review from Manuel Ferri in 2010. And he basically explained how by micropatterning cells, you can recapitulate some of the aspects of the native microenvironment, whether that's geometry, architecture, so connection with other cells, composition, mechanics, dynamics, etc. And so this is a immunofluorescence image of some mesenchymal stem cells on plastic. Just about everybody uses plastic, including us, for culturing cells, but it in no way reflects what a cell sees in the body, right? I mean, you have this rigid gigapascal synthetic material and this sea of nutrients sitting above the cells. And so with micropatterning, we can at least start breaking apart some of these things. And so here's a picture of cells that have been micropatterned in our lab in a range of different geometries. And the reason we do this is now, instead of just changing area, we manipulate some of the subcellular curvatures and aspect ratios and things like that. And we can get very different organization of the filaments within the cells. So this is a flower shape. And you see here we got the nuclei in the center, and we get this nice ring-like filamentous actin network. And it's just this beautiful web formation of microtubules throughout the cell. And by modulating that architecture, changing it up, using different shapes, you can influence the way that these things align. And use this as a method to study the importance of geometry and how it influences the cytoskeletal architecture. And so the platform we use to do this is microcontact printing on thin layers of gold. We like gold because it's such a well-developed chemistry for self-assembled monolayers. And so in essence, we make a PDMS stamp, so just a regular silicone rubber stamp. We can ink that with a chemistry of choice. In this case, we use this octadecane thiol molecule, which can chemisorb to the gold surface and form a very stable gold sulfur bond. And then by patterning that, we can then backfill the remaining regions outside with a non-adherent oligoethylene glycol moiety, which will prevent protein adsorption and cell adhesion. We can then physisorb to the surface of these hydrophobic regions some protein of interest. We usually use fibronectin, but we've also looked at laminins, collagens. And then, because we have these ethylene glycol regions, we can capture cells specifically on the islands that we stamped. And so we can do this across a range of geometries. This is some work that I helped out with during my postdoc, where we looked at motility features and contractility features using just a wide array of different geometries to see how you could influence, and these are with cancer cells, the migratory features of these adherent cancer cells by changing some of the geometric hues. And so the question we're asking is, can we utilize some of these to look at the influence that some of these more subtle cues have on differentiation? And so the first thing that we looked at was aspect ratio. And so using a similar protocol to what Chris Chen had devised in their 2004 work, we just took 2 bottles of media and mixed them 1-to-1. One would promote fat, one would promote bone. And when you do that on plastic, you get this crazy heterogeneous mixture of cells. You have little pockets where you get a lot of fat, a lot of bone in some regions, and it's quite variable. When you pattern the cells, you can just go across the surface and count cells that either express bone markers or fat markers or both. And there tend to be a small amount that presents both, usually less than 5%. And so when we did this with rectangles of the same area, but with increasing aspect ratio from 1-to-1, 3-to-2, and 4-to-1, And we counted the cells, we see an interesting trend that emerged, where that as we increase aspect ratio, we increase the proportion of cells in the culture that are turning into bone cells. And so we see in the square, we see about equal numbers of fat and bone cells, but as we go to this longer rectangle, we see more bone. And so we went to explore why this was, and one of the things that really stood out was as you increase aspect ratio, while keeping area constant, so there's no difference in spreading here, we see an increase in cytoskeletal tension, where now we get very large stress fibers along the long edge of the cell with more focal adhesions on the short end. And so similar to the spreading, we have an increase in actomyosin contractility in these geometries, which seems to be a cue to promote bone cell fate. Similarly, we did the same thing with a five-fold symmetric geometry, where we have a flower-shaped cell, and what we have here is these convex regions of curvature on the perimeter, and then a simple pentagon, and then this star-shaped geometry. And so, looking under a microscope, at first they all look the same, and so this was really startling when we first made this observation. And so when you expose them to the differentiation conditions, we find that going from this flower to a star shape, you see almost a complete lineage shift, where now on the star we see most cells are differentiating to bone cells. And consistent with the spreading and with the aspect ratio, when we do immunostaining for actin and vinculin for focal adhesions, we see that cells in the star geometry have a much higher degree of these bundle-like stress fibers between the points of the star. And the reason is that there's these non-adhesive spaces between these points where the cell has to overlap. And this is a cue to generate a lot of stress in the cells. And so they develop these strong stress fibers that are then anchored by very large focal adhesions, sometimes on the order of tens of microns, right on the tips of the cell. One thing that's really terrific about doing these pattern cultures is when you have unpatterned cells, they're all over the place. Every cell has a different shape. It's really tough to make any morphological conclusions about them. But when you do patterning, you're constraining every cell in the same geometry, so you can do cool things like make heat maps. And so what these are is we took 80 and 86 images of single cells that were stained for myosin 2A, and then overlaid them in ImageJ, a program you get from the NIH, and then you can generate an average immunofluorescence heat map, and which can give you somewhat of a semi-quantitative indication of a feature. And in this case, it's contractility, where we're looking at myosin 2. And you see that cells in this flower geometry have a diffuse myosin staining all across the cell, while in the star geometry you see a very high localization of myosin between the points of the star, which is consistent with this idea that it's in a more of a contractile phenotype. So we thought this was pretty cool, so we went to try to figure out what was causing some of this and whether we could attribute this directly to cytoskeletal tension. So we used some pharmacological inhibition studies. And so here's our control condition, where we get fat on the flowers and bone on the stars. When you treat these cultures with nicotazole, which inhibits microtubules and can lead to a high degree of cytoskeletal tension in the cells, but using a concentration where the cells remain viable and they stick to the shapes, we see that we get abrogation of this trend, where now all of the cells are turning into bone. And so we've lost this shape dependence by basically using a sledgehammer to knock the contractility up in these cells. And in contrast, you can do it with other molecules. So we inhibited myosin, a light chain, with blebistatin, and there we saw the same trend, but this time we see all of the cells turn into fat. And by blocking Rho kinase, which is an important player in the contractility of cells, we also saw abrogation of this trend in all fat. So this is consistent with our story, that these subtle geometric cues can influence actomyosin contractility, and by knocking it out, we can get rid of the shape-directed effect. Okay, one of the things that came out that was somewhat surprising from this is, and this happens when you run microarray experiments. Anyone who's done them will know this. You just get a big old barrel full of data, and you have to spend a lot of time digging through it. And so, I spent several months staring at microarray data, and there are a lot of pathways that seem to be differentially modulated by shape. But one thing that really stood out that I thought was interesting is cells in the star geometry had a high expression of these wingless type soluble proteins that are secreted, and they're involved in all sorts of stages of development, as well as cancer and bone cell formation. And we also saw increased expression of a lot of the mitogen activated protein kinase pathways that are involved in mechanotransduction, like the extracellular related kinases and C-Jun N-terminal kinases. So we were curious about what role they played in this shape-directed effect, and at the time, it wasn't so clear how these mechanochemical pathways influenced the lineage programs. So we wanted to try to figure out if we can get a direct link to gene expression. And so we inhibited Wnt extracellularly using just small protein molecules that inhibit Wnt signaling. So DKK1, which inhibits the canonical Wnt at the membrane. And when we add DKK1, we see a slight increase in adipogenesis and decrease in osteogenesis. Not very significant, though. When we add secreted Frizzle-related protein, which will block all Wnt in solution as well as at the membrane, we see a bigger increase in adipogenesis. And then when we mix those two, we see pretty much we lose all of our shape-directed differentiation, suggesting that with this contractility, the Wnt are really playing a serious role. And so the picture that I paint with this is that when you have a contractile cell and a cell that is not as contractile, the contractile cell upregulates a lot of the players that are involved in Wnt signaling. So the small GTPases, the rho kinase, and some of these pathways, because there's a convergence between many of these signal transduction pathways. So Wnt signaling to the membrane, where it reacts with these Frizzle receptors, will lead to an increase and an activation of rho kinase and some of the MAP kinases. And so we feel these pathways are working together to enhance this shape-directed differentiation. Okay? So, we thought this was really interesting, and so how can we utilize micropatterning to explore this further? And so we went back to this argument that the spreading of the cell influences osteogenesis. And I think a lot of biologists, and many I'd spoken with felt this way, still believe that paracrine signaling is very important and is likely to play a significant role in these different density cultures. And so to remind you, at low density seeding, you get a high degree of osteogenesis, shown here with the blue. Well, when you initially seed at high density, you get a low degree of osteogenesis. And Chris Chen and others suggested this was because of this mechanical aspect of spreading. So we wanted to look at paracrine signaling, and so the way we devised to do this, very simply, is by patterning cells at a different distance. So you can reduce the diffusional path length between the single cells. And so going from about 100 microns separating out every cell to 200 to 400 to 800. So they're nearly a millimeter apart in your dish. And so we did the analysis that way, and we dumped in the chemicals as before, and something really startling came out of this, where at very high density, we see most of the cells are turning into fat. But as we increase the spacing between them, we see an enrichment in cells that are differentiating to bone. And so this, my jaw absolutely dropped on this because it was unexpected, where we see at high density a lot of fat, but as you decrease the density, you get almost a complete switch here, where now most of your cells that are very far spaced apart are expressing bone cell markers. So clearly something's going on here. We're still struggling with what it is, but I'll tell you about some of the candidates. And so the first obvious thing to do, change the media. If there's some secreted factor that's being shot out from cells to their neighbors, if you take that media out, you'll influence whether or not the cells are seeing that. So we changed the media every 12 hours for a week. It was an interesting week. And what we see is hopefully expect to see, where here's our control condition where we see a decrease in fat as the spacing increases and an increase in bone. When you change the media every 12 hours, you now see more bone on every single shape. The problem with this experiment, of course, is that you're adding new osteogenic components in there, and there's a lot of other variables that could play a role. So it doesn't demonstrate completely solving this issue. So we played around some more. The next thing, of course, is take media from these high-density cultures. So we cultured cells at high density and took that conditioned media and fed it back. And we see a slight change in differentiation. So here's adding 1x media. So 1x is the 100 microns apart cells to 2x, 4x, and 8x spaced cells. And we see a slight increase in fat and decrease in bone, although it's not statistically significant, but maybe suggestive. And then we further took that media and ran some gels to see if we could isolate species. And actually spent quite a bit of time trying to fractionate the media to determine what could be causing this density effect, and which isn't much fun. It takes a lot of time, actually. And it's very messy, particularly when you got things like bovine serum albumin in your cell culture media, and you're trying to deplete that out and just mask this whole region of your gel. But we did see some interesting candidate species over here in the 30 to 40 kilodalton range, and they're interesting because of our microarray analysis again, where we saw that cells that were patterned had a very high expression of these secreted Frizzle-related proteins, which are Wnt inhibitors. And so we saw greater than 10-fold expression compared to a non-patterned surface with cells in these conditions. And so we did some Western analysis of our gels, and they're present and they increase over time. So these MSCs are secreting this Wnt inhibitory molecule over time. And so then what we did is we purchased some antibodies to block the secreted Frizzle-related proteins in these cultures. And what we saw is that here's our control again, more fat on the 1X that decreases as you increase the spacing. If you block secreted Frizzle-related protein 2 that we saw high expression of, no real change, maybe some slight change, but nothing significant. 5, nothing significant. But then here, with the secreted Frizzle-related protein 3, when you block those in culture, we see partial abrogation of this density dependence. So right now, this is one of our top candidates for a molecule that may mediate this paracrine signaling effect. Because here, when we block the secreted Frizzle-related protein 3, we see that pretty much all of our cultures, whether they're 1X, 2X, 4X, or 8X separated on the surface, are turning into bone. So we thought that's pretty exciting. We also supplemented that result by adding recombinant SFRP3. And this wasn't too surprising of a result because we had seen this with our previous work, where when you add this protein, it promotes fat cell differentiation. And so in our control condition, again, we see this trend. When you add recombinant SFRP3, you get rid of the trend, although you still see something going on here when the cells are really far apart. And so there's likely other factors that play a role here, but this is absolutely a candidate. And I think it demonstrates how, if you think outside of the box with some of these conventional micropatterning technologies, you can do some really cool fundamental science. All right. So I'm going to tell you now about a chemistry story, about how we can explore how ligands on a surface can influence stem cell fate. First, I'm going to have a drink of water. Getting parched. And so the hypothesis here was similar to the nanostructured surfaces, stiff matrices, geometry. We could utilize ligands that have an enhanced affinity for integrin receptors to change the cytoskeletal tension of the cell and therefore influence differentiation. And so to do that, we used our self-assembled monolayer chemistry, but this time using this maleimide functionalized monomer that we mixed into the SAM. And then we immobilized a thiolylated peptide, one which has the RGD peptide, shown here in red, which is the linear peptide that's utilized in most biomaterials proof of principle studies. We also used the cyclic analog that had been explored for years as a therapeutic candidate. And it's interesting because the difference between these is really just the conformation. And what this does is it's really a thermodynamics argument. And so when the peptide is in this bed conformation, it'll slide right into the integrin pocket, into the binding pocket. While if it's this linear peptide, the protein has to do the work and fold it over. And so because of this, you get higher focal adhesion turnover as the cells are grabbing onto these cyclic peptides. You tend to get more focal adhesions throughout the cell and greater spreading. And so we thought, if you get more focal adhesions, you get better spreading, maybe we'll get more bone cell differentiation. And so we used an array of gold islands for this, where each one has been modified with different peptides, so we can control within the same experiment. And this is the phenotype of these cells on linear peptide, where you see they're well spread, they've got lots of focal adhesions on the side. But when they're on the cyclic peptide or on fibronectin, which has a loop domain where the RGD is, so it's already in that bent shape, right? We see lots of focal adhesions, and many more than the linear case, and in particular on the interior of the cell. And they tend to be larger in area, have more cytoskeletal tension, etc. And so in our first experiments, thankfully, we decided not to add a bunch of media. We just let them grow, and then we fixed and stained them. And we got some really interesting results. So here we see our control, where with these heterogeneous populations of MSCs, anyone who works with MSCs has seen this. You always get a mixture of cells where some of them express alkaline phosphatase. And this is likely due to the heterogeneous nature of the culture, but also probably because some of these cells are becoming transit amplifying cells. They're moving more towards osteoblasts. And so we always get a mixture of these, but when you culture them on fibronectin for 10 days, we see that more of them are expressing alkaline phosphatase. When you compare the two peptide surfaces, both at high density, 1% of the surface has the peptide, or low density, 0.1%. On the cyclic peptides, we see a higher degree of alkaline phosphatase staining compared to the linear peptide. And that can be shown here, where we see for fibronectin in the high-density cyclic, the highest percentage of cells expressing alkaline phosphatase, which then drops off as you go to lower density and to the linear peptide. So this seems to suggest that perhaps the spacing and the affinity of the ligand for the receptor can influence the fate decisions. So we did some other staining. This is for RUNX2, which is a master osteoblast regulator. And we see a peak in the RUNX2 expression. And this is normalized nuclear to cytoplasmic fluorescence. And we see that for cells on the cyclic peptide and the fibronectin, we see higher nuclear expression of RUNX2, indicating that you have more of a proportion of lineage active RUNX2. So we also isolate RNA from this to verify our immunofluorescence results. And so we got RNA and we profiled these and found that we got the same sort of trend where here for alkaline phosphatase we see robust expression on cells on fibronectin, on cyclic peptide, but then a significant decrease in expression on the linear peptide. And a similar sort of trend we saw for RUNX2. And one of the things that was interesting with these experiments, and I started thinking about when I was actually considering some of Adam Angler and Dennis Disher's work, is the morphological changes that occur on these different materials. And if you put cells on gels, you'll see, as the cells can exert tractional forces differentially on these different materials, you can understand, and you see a similar thing here when you change the density of ligand. We're here on the cyclic peptides. Most of the cells have more of a cuboidal shape, very symmetric. But as you go to lower density cyclic peptide and the high density of the linear, you start to see elongation, where cells are getting more elongated. They're taking on different shapes. And then finally, at this very low density peptide surface, many cells start to look a little neuronal. And I'm not going to claim that these are neurons, but similar to what Adam Angler had shown, these processes being extended was one of the hallmark characteristics. And so I should point out too, when we quantitated these shape changes, the low density is really interesting, where we see that after 10 days in culture, we see a significant decrease in cell area as well as nuclear area. And when I see a change in nuclear area, that tells me something's going on with gene expression, and maybe the chromatin is condensing and some other program is going on. So we stained these for all sorts of markers. The result, I'll just give you the punchline, was that on the cyclic peptides, these cells would always tend to prefer to express markers associated with bone cell differentiation, while on the high-density linear peptide, we started to see expression of markers associated with myogenesis. So remember, on the intermediate stiffness gels that Angler and Disher worked with, they showed expression of myogenesis. And here at this high density of peptide that has a lower affinity for the alpha V beta 3 integrin, we started to see expression of muscle markers. And then these cells that were extending out all these processes on the low density, a good fraction of them were expressing beta 3 tubulin, which is a marker for neurogenesis. So this is that quantitated, where here you see it's not an incredible high amount, but it is significant. Here's the RUNX2 data showing that you get preferential bone marker expression, and then the MyoD on the high density linear peptide, and beta 3 tubulin on the low density linear peptide. We did RT-PCR of this as well, and we see the same sort of trends when we look at the gene expression level. And so we see higher expression in muscle markers and neuronal markers on these linear peptides. And so consider this example of stiffness, where people have been talking about for quite a while now, the importance of the material's elasticity and its stiffness in directing differentiation. So from this work, it suggests that in addition to stiffness, we may be able to modulate the density and the affinity of ligands on the biomaterial to get similar trends in lineage specification. And I think this is interesting for a number of reasons. For a practical reason, for biomaterials design. If you want to promote bone cell fate, have a high density of cyclic peptides. But also I think it's interesting if you reflect on the nature of the microenvironment that we're working with. If you're talking about brain tissue, you're going to have a soft material. The exposure of ligands for adhesion might be very different than if you have a very rigid calcified matrix, where you're going to have a lot more area for cells to grab onto, exert forces, and spread out. And so I think in many ways this result reflects that. In addition, one thing that has become a bit of a controversy just recently, that came out in a paper just several months ago, and I was at a meeting this summer where Adam Angler and some of the people, the authors of this paper, were there, and it got kind of heated. And that's exciting. It's exciting when science gets turned over and gets argumentative. And I think this has been in my mind and others for a long time, that when you cross-link polyacrylamide, which is the gel they used in this study, and you vary cross-linking density, you're not just varying stiffness, you're varying the porosity. And so this is an SEM image of soft gels and stiff gels across a range of mechanical properties. And so you get very different pore sizes in these materials. And if you're cross-linking your protein that the cells are going to grab onto at the surface, when you look at what the cell's going to see, it's going to see a very different distribution of protein depending on what surface it's sitting on because of this porosity effect. And so I think this is interesting in light of the result that we found, that by varying the spacing of these RGD peptides, you can vary the differentiation outcome. And so this may clearly play a role. And so this is the result that they found. This was a big collaboration that was done with Fiona Watt at Cambridge, as well as Wilhelm Huck and Joachim Spates, a number of these big names. And they did this, they published this paper where using polyacrylamide, and this time they used fat and bone. So fat is the soft tissue, bone is the stiff tissue. And they found that as you increase the stiffness, you get more cells, which is what others have found. But when in terms of differentiation, you get a decrease in fat and an increase in bone. So this is expected, that as on soft materials, you'll get more fat cell differentiation. On stiff materials, you'll get more bone cell differentiation. And so this is consistent with this argument that there's lineage matching between the differentiation outcome and the mechanics of the material. But then they went and used PDMS, which doesn't have a porosity component. And they showed that when they modified PDMS of different mechanical properties, that they saw no difference in the number of cells, no difference in adipogenesis, and no difference in osteogenesis. So here they have different mechanical properties of their substrates, but the same ligand density, the same amount of immobilized protein, and they see no change. So this is pretty controversial. There's a number of problems with the experiment, I think, in terms of the PDMS material and what they're actually detecting, but I think it's exciting, right, because it opens up new areas for discussion. In terms of this question, I'm sitting more here, where I think that clearly these things play different roles. So this was a paper done a couple years ago with Fu and Chris Chen at Penn again, where they used these elastomeric micropost arrays that they have. And they did a really cool trick where they just changed the height of these posts. And so the mechanical properties of the material is the same, but they're just changing the height such that the stiffness that the cell sees is different across these. And so what they showed is here cells on very short posts spread out, and they tend to differentiate into bone. Cells on these long posts that are very flexible tend to ball up and they turn into fat. But here, arguably, the adhesion ligand presentation is going to be the same across these surfaces. And so I think this is still open debate, but I think it's definitely an interesting area. Okay? How are we doing on time? Okay, still got a few minutes. All right, so now I'm just going to show you some of the new things we're doing with combining these cues. And so, as I mentioned at the start of my talk, I think the next generation of 2D assays is going to be involved, now that we've got lots of control over microstructure interfaces, surface chemistry, is looking at how these things play together. And shape and stiffness is one that I've been interested in recently. And so this is the idea. We can now vary things like shape and stiffness. We can also look at variations in ECM chemistry and stiffness and shape. And so my lab's actively looking at ways that we can develop platforms to vary these things rationally and controllably. And shape and stiffness, I got excited about this. Here's the work by Angler and Disher again, where we see very different shapes across these different stiffness gels. Here's a neuron, myoblasts, and osteoblasts. But when I did these experiments, I saw a mixture of fates. And so I just showed you some work where they showed on soft gels you get fat cell differentiation. When we've cultured MSCs on these very soft gels, we see that some cells express beta-3 tubulin, a low percentage of them, while many cells start to accumulate lipid droplets, which appears that they're going towards an adipocyte fat cell fate. And similarly, on stiffer materials, you tend to get a mixture of cells that are expressing markers of muscle and bone. And so the question is, can we devise a way to exclusively promote these different fates on these hydrogel materials? And so one way, perhaps, is to use shape, since there's so very many different shapes between these cell types. And so we adapted a technique, and this is work done by a student in my group, Amr Abdeen, And this is a technique developed many years ago, actually, where polyacrylamide gels are modified with this hydrazine treatment, and then you oxidize a glycoprotein to have an aldehyde that you can then immobilize to the gel. And we developed a strategy to robustly do this across a range of stiffnesses. And it was really tough to get it on the soft materials, but we are able to pattern cells. These are single mesenchymal stem cells, pattern on gels with 0.5 kilopascal of stiffness. And so we decided to see what patterning would do. Here's an example of sort of an interesting early development that we're currently investigating, where we found that when in unpatterned culture, in pattern culture, we see very different mixtures of these lineages. So here, when cells are patterned, it's tough to see here, but that's because the marker isn't really there. But these are patterned cells, and they show very low levels of beta-3 tubulin. While in unpatterned culture, a subset of these cells will elongate, develop these processes, and start expressing this marker for neurons. And this is that quantitated with this box and whisker plot, where we see throughout the spread, there's some of these cells that are expressing higher levels of beta-3 tubulin. Maybe more interestingly, when we pattern cells and we stain them for adipogenesis markers, we see that most of the cells across the surface are expressing markers associated with fat. And so I think this is really interesting. In a couple experiments, we saw greater than 90% of the pattern cells had lipid droplets. And my hand-wavy argument is right now is that perhaps what we're doing is limiting some of these morphological changes that occur on these different materials of different stiffness, where if a cell needs to elongate and form dendritic processes in order to undergo this neurogenesis pathway, and it's restricted by being patterned in these small shapes, perhaps it can't do that and it chooses fat sulfate. And so perhaps this could be a platform to modulate between these different lineages. And so we're currently exploring this in other systems to look at other differentiation outcomes like muscle and bone, and perhaps finding ways that we can guide neuronal process extension. On stiffer gels, this is just a snapshot of some recent result from a student in my group, Jun Min Lee. And what Jun did is he used this shape that he calls affectionately the Batman shape. He actually sent me a picture for our calendar that has like Batman standing on a tower and the light is showing this cell up there. I should have put that in the slides. But the idea of this geometry isn't because we like Batman, it's that it has some of these cues that we've seen previously on gold to promote bone cell differentiation. So it's got aspect ratio, and it's got regions of subcellular curvature. And what Jun found is when he leaves cells just to sit on 10, 20, 30, or 40 kilopascal gels, he sees expression and then an increase at 30, just like Adam Engler and Dennis Disher did, where this seems to be an optimal microenvironment mechanics to promote bone cell fate. But when cells are patterned in this Batman geometry, you see a higher expression of RUNX2, this regulator of osteogenesis in this geometry. And so I think this is a pretty cool result, and we're going to try to find ways that we can do this for myogenesis and these others in order to maximize the lineage outcome. And the idea in the long run would be able to control across a material's surface differentiation spatially in the way that you want, so that you could have gradients of cells. Because remember, there's no chemicals added here. This is just the mechanics and the geometry of the cells. So you might be able to pattern fat and bone, and integrating it with our ability to use microfluidics and some of these technologies to form gradients of stiffness. You could envisage all sorts of really cool applications. All right, I'm pretty much done here, aren't I? I'll just go really briefly through this. This is just a news story that's come out that I think is interesting, and it revolves around this idea that kind of came a bit out of a serendipitous discovery, where we patterned cells and they were left, and these chemistries on gold are not so stable. And so cells can migrate and leave them after about a week. And these were left for too long. And so they migrated out. And they were fixed and stained anyway, thankfully, because we saw something really interesting, where when cells, as we culture them normally, are allowed to express alkaline phosphatase when they divide, when they were patterned, after they escaped, they're no longer expressing alkaline phosphatase for whatever reason. And so we're thinking this could be related to quiescence. And so we did some staining to see if these cells are synthesizing DNA, and when they're patterned, they tend to not. If they're in very small patterns, these cells seem to be turning into a quiescent state. And then one thing that I think is really exciting that's coming out is we see that in these patterned cells, we're seeing higher expression of stem cell markers. And so we did these experiments where we patterned the cells, And then after they escaped, we trypsinized them and then reseeded them at the same density and looked at stem cell markers. And here for endoglin and STRO1, we see a higher expression of stem cell markers in the cells that had been patterned. And importantly, we see high expression of alkaline phosphatase in the unpatterned cells. So this might be a way to prolong the stem cell phenotype in culture. And so you can imagine being able to do this to maintain that. And I think it's pretty neat work. And this is work by Doug Zhang in my group. And they also can still differentiate. I'll show that before I close here. And so here, this is alkaline phosphatase staining. You see less differentiation, and that's presumably because many of the cells in the starting population are still mesenchymal stem cells and not progenitors. And we see this with fat as well. Okay, I'm just going to quickly summarize. Cell geometry and these things influence each other. This is really at the heart of our group's work and seeing how we can use these platforms together and combine these cues to maximize outcomes. And in this new work, I think is pretty exciting, where we might be able to use patterning to prolong this MSC phenotype. And with that, I'd like to thank my group. This is an older picture, but we've expanded a bit. In particular, I want to thank Amr, who's developed a lot of the hydrogel chemistry, June, who's done some of the patterning on hydrogels, and Doug with this recent story. I've got to thank Milan Merksic and his group, of course, for some of the preliminary work that I did during my postdoc, and these collaborators listed here. With that, I'll be happy to answer any questions. Thank you.
Any questions from the group? Very clear.
Yes.
I was really interested in what you mentioned about the porosity of the polyacrylonide and then maybe having a role in this sort of stiffness induced differentiation.
Yeah.
But you said some of the studies that were the one study was kind of countered that argument, had some flaws. I'm kind of curious if you could elaborate a bit more, maybe?
On the flaws of that study?
Or just, yeah.
I think, well, one of the main is that they only looked at the fat and bone cell differentiation. And so I think before we can relate that directly to a lot of the work that's been done with muscle differentiation and things like that. I also think that the techniques they used for measuring the stiffness of the materials, I believe they used AFM, some of these things, which are valid, but it would have been nice to see some complementary approaches. And also, they didn't characterize as well as maybe they could have, how the cells are interacting with the surface. So I mean, from a surface chemistry perspective, I'm interested in how the cells are actually grabbing onto these ligands. So I would have liked to see a more detailed analysis of focal adhesions and tractional forces and things like that. So those types of things. I'm sure there's other people on this campus that have many other issues too. Yes?
So other than maybe shape, if you're just looking at imaging of the cells, are there other sort of markers that differentiate different stem cell populations or things? Did you see anything like dynamics or time to the structure or any other sort of physical or morphological parameters that you could use perhaps one day in vivo to differentiate the cells?
Oh, excellent question. I mean, that's something that I'd love to do. We haven't done any of that. Most of our analysis has been static and measuring large populations of cells. But I mean, clearly, and I think the field's moving in that direction too. We're doing some live cell analysis, and I started a collaboration during my postdoc with Pravis Mogi at Rutgers. He does a lot of this high content imaging of cells, and looking at things like cytoskeletal descriptors is something that I think would be really exciting. Where after a day or two, you might be able to see rearrangement of the cytoskeleton and be able to make more firm correlations between these morphological changes and outcome. I mean, it's absolutely exciting, but I haven't done it before.
Questions? I have a quick one. I mean, you give a really nice overview of all the basic science related. What are the important parameters, perhaps how some of them correlate in ways. What's sort of the state of the art then in terms of materials where you can translate a lot of these things into maybe therapeutic devices? Are there things that are occurring that allow sort of programmable matrices that would maybe allow some of the parameters you're talking about to be?
Sure. Yeah, no, I mean, I think it's we're pretty new at being able to do it robustly, but absolutely, there's new hydrogel chemistries coming out all the time. Christy Ansett is one of the names that pops in, where she can use 2 photon techniques to locally polymerize gels in a 3-dimensional way that maybe tissue shape than a 3D material. And a lot of the control over like some of these peg-based multi-arm gels where you may be able to change the length of the pegs and rationally incorporate peptide ligands into some of these scaffolds. So synthetic scaffolds, I think we're starting to develop cool tools. Whether, I think we're a long ways off before we can translate those into something that might be biologically relevant, but maybe not too far. Maybe not too far. That's my feeling of the state. Making progress. I don't think we'll be able to fabricate a scaffold that resembles what happens when you decellularize a heart, for instance. But I think we're learning some of the principles, and these fundamental studies I think feed into that, where we can, at least with these synthetic strategies we're developing, we know that we can use materials principles to vary things like stiffness and ligand presentation.
All right, let's thank Chris once again.